Hello everyone, my name is Eric Swenson, and today I'm going to talk about testing the addition of silver nanoparticles to existing silk fibroin scaffolds used in bone tissue regeneration for antimicrobial properties and improved promotion of osteogenesis. This presentation will cover the motivation, background, state-of-the-art, knowledge gap, strategies, and end with a summary of important takeaways. Musculoskeletal tissue injuries and diseases can be difficult to treat and can have serious health effects. This is becoming increasingly important because as the world population continues to grow, so does the amount of people who are affected. Bone tissue engineering provides a very promising solution. Osteogenesis, the formation of bone, can be facilitated by specific biomaterials and methods. I will focus on the design and engineering of cell scaffolds for this purpose. This technology is held back by the fact that a significant portion of failed attempts that occur when using this technology in vivo is due to bacterial infection. Silver nanoparticles have antibacterial properties and promote osteogenesis. Silver nanoparticles have been used for their antibacterial ability in several ways, including the health industry, cosmetics, textile coatings, and food storage. In orthopedic devices, antibacterial properties are very useful, as bacteria have been shown to form biofilms that resist the immune system and antibiotics. Silver nanoparticles have been widely demonstrated to kill gram-negative and gram-positive bacteria, but also benefit from the fact that they've been shown to be less likely to cause resistant bacterial strains. In addition to bacterial properties, silver nanoparticles promote osteoblast differentiation in vitro and promote bone fracture healing in vivo. Both of these properties of silver nanoparticles make them potentially useful in the field of tissue engineering. This figure shows results from a previous study. They observed the effect of silver nanoparticles on osteogenesis, and focusing on mouse mesenchymal stem cells, they noticed increased alkaline phosphate enzyme activity, especially at 14 days, which is an indication of osteoblast differentiation. Cell scaffolds facilitate tissue regeneration. Many different types of materials have been considered and extensively researched for this application. This includes metals, natural polymers, composites, and ceramics. Natural polymers especially have potential for tissue regeneration because they best mimic the natural extracellular matrix. Some examples are chitosan, polylactic acid, collagen, gelatin, and silk fibroin. Scaffolds also need to have strong mechanical properties, high biocompatibility, a specific rate of degradation, specific porosity, and many other characteristics to carry out their function. This image shows that when scaffolds are used in combination with stem cells and additional molecules, such as growth factors, engineered tissue can result. Now that you understand the motivation behind this proposal and background information, I will next go over the state-of-the-art and the knowledge gap. Silk fibroin-based scaffolds have become a leading candidate for bone tissue engineering. Silk has been found to have highly optimal biocompatibility, mechanical strength, and degradation rate that makes it rank above other materials for bone tissue engineering. The versatility of silk scaffolds mean that they are also capable of being further engineered and tailored through the use of additives, composite materials, embedded minerals, and nanoparticles. Existing bone tissue scaffolds need to be engineered to lessen the risk of infection that comes with this type of therapy. This is important to consider because cell scaffolds need to be in place in vivo for an extended period of time. The development of a significantly effective prevention method remains to be accomplished. In order to fill this gap, existing bone tissue scaffolds should be engineered to fight infection. This could be done by improving the antibacterial activity of the scaffolds or by reducing the time required for regeneration. The addition of silver nanoparticles would be a relatively simple solution and would be added during the preparation of the scaffold. A study was done where magnesium oxide nanoparticles were incorporated into silk fibroin scaffolds to improve hydrophilicity. Integrating silver nanoparticles into these scaffolds could be done in a similar fashion. Next, I will explain strategies to test and analyze the effectiveness of silver nanoparticles when incorporated into bone tissue engineering technology. The first aim will be to characterize the antibacterial activity of silver nanoparticles when they are incorporated into silk fibron-based scaffolds. This could be investigated in vitro on small-scale silk fibron-based scaffolds prepared with and without silver nanoparticles. Other methods to prevent infection would also be tested and compared, such as antibiotics. The bacteria used in this experiment would be E. coli due to their well-known characteristics. The results would be analyzed based on the concentration of leftover E. coli after a certain amount of time after treatment with silver nanoparticles, antibiotics, or neither as a control. The next step of this aim would be to graduate to silk fibron scaffolds in vivo using mouse models and testing for the same specifications. The second aim of this study would be to measure the degree to which silver nanoparticles promote proliferation and differentiation in vivo using mice. This strategy would be partly based off an in vitro study done where mRNA Illumina sequencing was done on total extracted RNA on several days of the experiment to assess the effect of silver nanoparticles in vitro on gene expression in osteoblastic cells. This is shown in the figure here. Their work found that silver nanoparticles increased expression of genes relating to cell differentiation and proliferation. A similar experiment could be done by instead seeding cells on silk fibron-based scaffolds with and without silver nanoparticles, and then comparing the difference of proliferation, differentiation, and gene expression between the two. This would be important to do because the interactions between the cells, the scaffold, and the silver nanoparticles could lead to unexpected results. A third aim of this study would be to determine the effects of concentration of silver nanoparticles in the scaffold to optimize for effectiveness while also reducing potential toxicity that could occur due to high concentration. This could easily be done as an extra step to the first two experiments, but could also be done separately to help understand any interactions between the silver nanoparticles, the silk fibron-based scaffold, and bone tissue if done in vivo. A previous study was done to test the effect of silver nanoparticle concentration on mouse mesenchymal stem cell proliferation. The results are shown in the top figure, with concentration on the x-axis and cell proliferation on the y-axis. There was a significant increase at 4 micromolar. Another study covered the long-term effects of exposure to silver nanoparticles. The results are shown in the bottom figure, with concentration on the x-axis and percent of cells left over on the y-axis. They noticed non-reversible impairment of cell viability when the concentration was around 25 micrograms per milliliter. These values would be used as reference points for the proposed experiments. Silk fibron scaffolds are commonly used in the field of bone tissue engineering and are ideal for further engineering. A current issue with this technology is that bacterial infection can interfere with tissue regeneration. It is important because an engineered silk fibron scaffold with full resistance to infection has not yet been developed. Silver nanoparticles are a potential solution to issues due to infection because of their well-known antimicrobial properties and potential in accelerating the cell proliferation and differentiation process. The experiments outlined here would allow for better insight into the effect of silver nanoparticles on the antibacterial and osteoinductive properties of these scaffolds in vitro and in vivo. This information could then be referenced by later studies with a similar strategy. In general, improving the safety and efficacy of this technology would allow for it to become clinically available for humans in the future. Thanks for watching, and I am happy to answer any questions.